Доброе утро, Расул Салихович.
Доброе утро. Доброе утро. Я вас по нику не узнал. Думаю, это Роше. Так, сейчас я вас сделаю соорганизатором. Ага.
Почему-то у меня Рош. Я не знаю, надо как-то это исправить.
Вот я думаю, Роше даже, если по-французски Роше. Наверное, компания какая-то. Сейчас ребята подключают.
Наверное, я когда-то... Да. Сейчас я вот делаю share.
У нас ВКонтакте почти 12 просмотров. В этом... О, 13. Такой большой идет рост.
А так видно мои слайды?
Да, все замечательно видно.
Хорошо. И меня полностью видно целиком, да, лицо? А то мне кажется, что у меня только половина лица.
Нет, все, слайды все видно.
Хорошо. Ждем.
Ждем ребят. Вот они, я что-то вот сейчас по монитору смотрю, они почему-то это через социальные сети заходят очень много. Через через Zoom не так много.
Может быть, через Facebook? У меня высветилось на Фейсбуке.
Да, на Фейсбуке сидит 11, на ВКонтакте сидит, смотрит 11, и в Ютубе 20 человек, и у нас здесь 10. Но они сейчас будут подключаться, еще время есть, ссылку-то мы только 10 минут назад ребятам послали. Поэтому, наверное, немножко подождем. Я сейчас выключу звук у всех,
Конечно.
кроме вас, а вы потом себе звук включите. То есть вы сможете включить, ребята не смогут.
Да, я включила.
Угу. У нас еще 4 минуты. Да, в социальные сети очень так это активно ребята заходят, смотрю. Dear students, you can add yourself to Zoom, as well as you can watch this lecture through social media like YouTube, VK, Facebook. And I see most of you prefer social media instead of Zoom. Okay, this is normal. Так, ну еще 3 минуты, пока поразвлекаем. At the end of our school, we will give you some certificates for those who will successfully pass some knowledge assessment after when we finish the school. Okay. Может, начнем потихоньку, то есть вы с вступительных слов, а ребята пусть добавляются, потому что у меня тут звонков куча идет. Я параллельно, чтобы и вас слушал, и работал.
Хорошо, Бог в помощь. Я просто еще через 2 минуты начну, потому что ровно по времени, чтобы было.
Ага, добро. Я тогда себя звук отключу, мне тут на подпись принесли, и буду с удовольствием слушать про вакцины. Меня ждет ревакцинация, я думаю, скоро. Ребята наши, кстати, прививаются очень интенсивно. Сейчас уже на сегодня 88 привившихся. Часть
Бог на пользу.
В своих странах. Ну, нормально. Я думаю, после вашей лекции будет 800 желающих привиться. И что самое интересное, мы надеемся, что в России послушают эту лекцию, и наконец-то у них сознательность появится, они пойдут вакцинироваться. Потому что иностранцы, парадоксально, да, иностранцы хотят вакцинироваться, а россияне, которым на блюдечке вакцины, вакцинироваться не хотят. Вот это парадокс.
Да. Человеческая натура.
Да. So, dear students, soon we will start our lecture shortly. Naila Rafaelna, the floor is yours. Good luck, and I hope our students will get very, very interesting and encouraging information regarding to vaccination.
Thank you very much, Rasul Salihovich. It's almost exactly 11 a.m. and it's time to start our second lecture. Lecture number 2 will be about the vaccines used against the COVID-19. For those who did not listen me at the first lecture, I would like to introduce myself again. I'm Professor Naila Bulatova, and I'm MD PhD, and I work at the School of Pharmacy at the University of Jordan, Amman, Jordan. As you know, the pandemic of COVID-19 started, or actually the first outbreak started in December 2019, and the genomic sequence was already published after 2 months. On January 11th, 2020, it was published, and this allowed the research and collaboration among scientists and biopharmaceutical manufacturers quickly started working on the vaccine. As you know, various countries try to produce their vaccines, and they use various methods for the discovery and manufacturing the vaccines. First of all, many of you may ask me a question. Is vaccination recommended in previously infected individuals? Some of you may believe that if you have sustained COVID-19, it is not necessary to do the vaccination. However, one of the authorities, worldwide recognized authorities, which is the CDC. CDC stands for the Center of Disease Control and Prevention, the major body in the United States that takes care of the infectious diseases. So the CDC currently recommends vaccination for individuals who have already had COVID-19 and recovered. And they didn't even determine the period after which the infected individual can receive the vaccine. The period is not defined, which means that after recovery, very soon, the vaccine can be received. And this is because the vaccinations provide a safer and more reliable way to build antibodies compared with the infection itself. If you measure your antibody titer after the infection, you find it is quite low compared with the post-vaccination antibody level. Patients may receive the vaccines once they have recovered, as I said, from the acute illness. Actually, if he was symptomatic, because many people are asymptomatic, as we know, and meet the criteria to discontinue isolation, usually after 3 weeks of the onset of infection. If the course was not severe, the vaccination can be done. However, those people who receive treatment for the COVID-19 with monoclonal antibodies or convalescent plasma should wait for 90 days before receiving the vaccine. And I would emphasize again and again that the current clinical evidence shows that vaccines provide substantially higher protection against COVID-19 infection compared with the immunity from a previous COVID-19 infection. So from this information, we can conclude that even previously infected individuals should undergo vaccination against COVID-19. For example, the mRNA vaccines, or actually people who receive these vaccines, we call them vaccinees, people who receive the mRNA vaccines have higher antibody titers, up to 10 times higher than convalescent plasma from donors who recovered from natural infection. Which means that the scientists compared the titers of antibodies after the disease with the titer of the antibody after the mRNA vaccines, and it was 10 times higher in the latter case. Early studies found vaccination of patients with prior SARS-CoV-2 infection enhances not only the antibodies response, but also T-cell immunity and memory B-cell response, and neutralizing antibodies effective against emerging variants. Emerging variants are those new variants that are discovered nowadays, like Delta, Lambda, etc. So again, I would emphasize the importance of vaccinating both uninfected and previously infected persons to elicit cross-variant neutralizing antibodies. As for the methodology of vaccine development, several platforms are used, several types of vaccines are used. As you have heard in the news, there are RNA vaccines, there is inactivated virus, there are viral vectors, protein subunits. All of these platforms are available for the vaccine development. And the variety between the vaccines is in the number of doses. Some vaccines, they require 2 doses. Other vaccines, 1 dose is enough for them. And now it is a debate that for protection against the new variants of the coronavirus, there might be a need for the third so-called booster dose of the vaccine for those who receive 2 doses after some time, after half a year. Usually some authorities recommend the third dose, but this is still under the debate. Other authorities say that it is not necessary. So it depends on the type of the vaccine, the number of doses, and also the speed of development varies. Some vaccines, it is very easy to produce and to produce the industrial amounts of the vaccine. And this is reflected on the scalability, how many people can receive this vaccine. And other variables that increase complexity of distribution of the vaccines include storage requirements. You know, some vaccines, they require minus temperature, means being frozen. Other vaccines need refrigeration, and also whether more than a single injection is required for optimal immunity. Let us see a very interesting video or animation, how different coronavirus vaccines work. Let me just allow the sound. Hopefully you will hear the sound.
During menopause, estrogen drops and cortisol rises, causing unwanted tummy fat. And running
This is
an hour a day or cutting calories super low is only going to make the tummy worse. Hey.
During a viral infection, the virus attaches itself to a human cell using its spikes. Following entry to the cell, it uses the host cell's machinery to replicate, producing viral proteins and genetic material. These are then assembled into new viral particles, which are released as the host cell dies. New viral particles again infect more cells, destroying body tissues, producing symptoms. Infected cells alert the immune system by displaying pieces of viral proteins on their surface. By doing so, infected cells are said to present the viral antigen to certain immune cells, namely cytotoxic T cells, and thus activate them. At the same time, debris of dead cells and viral particles themselves are picked up by so-called professional antigen-presenting cells, of which dendritic cells are most effective. Dendritic cells patrol body tissues, continuously sampling their environment for intruders. After capturing the antigen, dendritic cells quickly leave the tissue for the nearest lymph node, where they present the antigen to another group of immune cells known as helper T cells. viral particles also activate B cells in lymph nodes. These groups of immune cells work together to mount two types of immunity specific to the viral antigen: cell-mediated immunity and antibody-mediated immunity. The process, however, may take up to a couple of weeks, during which time the person is sick and the virus will have destroyed a vast number of cells. Vaccines deliver viral antigens to trigger immune responses without causing the disease. The events of a vaccine-induced immune response are similar to that induced by a natural infection, although some types of vaccines may induce only antibody-mediated immunity and may therefore be less effective. The immune process can sometimes produce symptoms similar to a mild infection, even though there is none. As the lymph nodes near the injection site start producing antibodies, they may become swollen and tender for a few days. These are signs that the vaccine is working. Many existing vaccines contain a weakened or inactivated virus. Because the whole virus is used, these vaccines require extensive safety testing. Attenuated vaccines may still cause disease in people with compromised immune systems. Inactivated vaccines only induce antibody-mediated immunity.
But as I said,
Subunit vaccines contain only part of the virus, usually a spike protein. These vaccines cannot cause disease, but they may not be seen as a threat to the immune system and therefore may not elicit the desired immune response. For this reason, certain substances, called adjuvants, are usually added to stimulate the antigen-presenting cells to pick up the vaccine. A subunit vaccine may also consist of empty virus shells without genetic material. Having a typical size and shape of a pathogen, these vaccines may not require adjuvants to be perceived as danger, but they can be difficult to produce. Nucleic acid vaccines contain genetic information for making the viral antigen instead of the antigen itself. DNA vaccines introduce viral DNA into the cell's nucleus, where it is transcribed into mRNA. The mRNA is translated into viral spike protein in the cytoplasm. The protein is then displayed on the cell's surface, just like with other types of vaccines. Naked DNA vaccines require a special delivery method to reach the cell's nucleus. Alternatively, a harmless, unrelated virus may be used as a vehicle to deliver the DNA. In this case, the vaccine is also known as a viral vector vaccine. For example, the Oxford AstraZeneca COVID-19 vaccine uses a chimpanzee adenovirus as a vector. The adenoviral genome is modified to remove viral genes, and the coronavirus spike gene is added. This way, the viral vector cannot replicate or cause disease, but it acts as a vehicle to deliver the DNA. The reason a non-human adenovirus is used is because most people may have been exposed to human adenovirus and have immunity against it. The immunity could destroy the vehicle before it can deliver the DNA and thus blunt the vaccine's effectiveness. DNA vaccines have raised concerns about the possibility of viral DNA integration into human genome. However, studies in animal models have shown that integration frequency is well below the frequency of natural, spontaneous gene mutations. mRNA vaccines introduce mRNA that contains information for making the viral protein. mRNA molecules are delivered within a lipid covering that will fuse with the cell membrane. Once inside the cytoplasm, the mRNA is translated into viral antigen, which is then displayed on the cell surface. Unlike DNA vaccines, mRNA vaccines are extremely unlikely to integrate into human genome, but they are fragile and require uninterrupted cold chain for transportation and storage.
So they need special storage in the freezer. Okay, let me continue. Talk after such a brief overview of the vaccines, different types of vaccines. before talking about each of the vaccines, I would like to stress that some methods are all traditional methods, for example, using the inactivated virus, but for example, mRNA vaccines, they are quite new and they were previously developed and tested in animals against influenza, rabies, and Zika virus. And the following table compares different types of vaccines. So now we know that we have 4 different types of vaccines, major different types of vaccines. mRNA, based on the ribonucleic acid. It was developed fast, but low to medium manufacturing scale. That's why many countries still cannot receive this vaccine. It requires 2 doses, 21 days apart. And the major representatives of these vaccines are Pfizer's BNT162b2 vaccine or BioNTech. And the second one is mRNA 1273 by Moderna. Both are from the United States. The DNA vaccines can develop fast, but manufactured at medium speed. They also require the 2 doses, and it is still investigational Inovia vaccine. While another type of DNA vaccine is viral vector, where the adenovirus is used as a vector, as I told you, and it is developed at the medium speed, but can be manufactured at a high scale. Some of them require just 1 dose, like Johnson & Johnson vaccine, the American vaccine, also called AD26-CoV2S. And others, they require 2 doses, such as AstraZeneca, which is abbreviated as AZD1222-AD5-CoV. Also, we refer here the Russian Sputnik from the Gamaleya institution, Sputnik V. It is also the adenoviral vector vaccine, not presented here because this is some sources do not mention the Russian vaccines. The protein subunit is contained in the Novavax vaccine, abbreviated as NVX-CoV2373, medium to fast development, high manufacturing scale, and the Russian vaccine is Epivax Corona, which contains the peptides actually as protein subunits. And a very traditional way of producing the vaccines is to use the inactivated virus. The whole virion particle is used, it is killed, and we are able to quickly produce large amount of vaccine, and we require 2 doses. Examples produced, presented here are Covaxin, Ocugen, and Parada Biotech. I think this is Indian vaccine, abbreviated as BBV152. Also here we have the example of Russian vaccine by Chumakov Institution. I will talk about these vaccines later. And also a Chinese vaccine, which is Sinopharm. By the way, I was vaccinated by this vaccine in January 2021 in Jordan. And actually this vaccine, I think almost or more than 1 billion doses were given. So maybe the largest number of people on the planet, they received this Sinopharm vaccine from the inactivated virus. Now, I will characterize each of the vaccines in more details, starting with the Western vaccines. And the first type of vaccines I will cover will be mRNA vaccines. Here we have Pfizer, BNT162b2, which is a nucleoside modified messenger RNA vaccine that encodes an optimized SARS-CoV receptor binding antigen. You remember the receptor binding domain, RBD, of the S protein or spike protein of the virus I was talking about at the previous lecture. It is responsible for the binding to the target, which is ACE2 receptor on the surface of the cell. So this mRNA encodes for the fragment of the S protein. So since it is encoding for this protein, when the vaccine is given, the S protein is produced by the host, by the cells of the host, and this presence of S protein, as if we have the virus in our bodies, it elicits the immune response, as we saw in the video. As I told you, Pfizer vaccine needs 2 doses 21 days apart. The efficacy was 95% against the original strain, which is the Wuhan strain, at 7 days after the second dose. But what is important, you see, when we are talking about the efficacy, 95% means that still there are 5% that may be infected. But the point here is that there is 100% protection against severe disease. So you might still have a chance of being infected, but your disease will be mild to moderate. But there is 100% protection against the severe disease. Regarding the adverse effects, most of them were mild, and the only grade 3 adverse event was fatigue at about 4% of individuals, and 2% of individuals complained of headache. The most common reaction, however, was short-term mild to moderate pain, while severe pain occurred only in less than 1% of individuals who received this vaccine. Second mRNA vaccine, also American one, is the mRNA 1273 Moderna vaccine. It also, this mRNA also encodes for the S2P antigen, and it also needs the 2 doses, but the interval between them is 28 days, not 21 days. And overall efficacy was about 94% for the original Wuhan viral strain, and it was greater than 95% effective against the severe disease. Now, because the new variants develop of the virus, and the Moderna conducted additional investigation, and they have shown that after receiving the 2 doses initially, as I told you previously, now the 3rd dose may be administered, which is a single, we call it booster dose. It increased the neutralizing titers against SARS-CoV-2 and 2 variants of concern, which are B1351 and B1, in previously vaccinated clinical trial participants, which means that the 3rd dose might be necessary to protect against these new mutants or variants. And when we compare between the booster doses of mRNA 1273 and mRNA 1273.351, the second one achieved higher titers against the variant of concern, which is B1351, than a booster dose of another variant or another type of vaccine, which is mRNA 1273. So the evaluation of these boosters is still ongoing, and as I told you, it is not very well established yet whether we should receive the 3rd boosters of such vaccines or not. Probably you have heard or you have seen in the social media that one of the concerns with the mRNA vaccines is the myocarditis and pericarditis as the side effect. The first information appeared in May 2021, and there was a case series of 7 adolescent males. For some unknown reason, the males are affected more than females, and the young people are affected more than the older people. They presented the symptoms of myocarditis after a few days, 2 to 4 days, let us say, after vaccine administration, particularly after the 2nd dose. And the diagnostic tests showed the elevated troponin, ST elevation, and diffuse myocardial edema. However, none of these vaccine recipients were critically ill, and all responded quickly to treatment. The treatment consisted with non-steroidal anti-inflammatory drugs, and several people also received glucocorticoids such as prednisolone. So this data were reported to the Vaccine Adverse Effect Reporting System, abbreviated as VAERS, following approximately 300 million mRNA doses administered through June 11th, 2020. Total, so the number of these myocarditis cases were 1,200 something, which means that among 300 million, we have this prevalence of myocarditis, which is a very, very rare adverse effect, maybe 0.3 or less %. And most of them, I repeat, appeared after the second dose, and nearly 80% of them were in males. And still, despite of this incidence of myocarditis, pericarditis, the CDC, the authority that we have mentioned previously, and also American Academy of Pediatrics, stress the benefit of the vaccine at preventing severe COVID-19 disease, hospitalization and death, and recommend vaccination. What I'm trying to say here, okay, we have the adverse effects from some vaccines and some treatment, but when you always weigh in medicine harm versus the benefit of protecting against severe COVID disease, hospitalization and death clearly exceeds the risk of having the myocarditis, which is not life-threatening, and in most cases it is mild and treatable. And this is a very rare adverse effect. So the CDC instructs currently the patients who receive the mRNA vaccines to promptly report if they have chest pain, dyspnea, or palpitations after receiving the vaccine, because this may be the indication of myocarditis and pericarditis. And as I said, the treatment of this complication of vaccination consists of non-steroidals, glucocorticoids, and also intravenous immunoglobulin IVIG. Additionally, if someone is engaged in athletic activity and physical activity during the myocardial injury and inflammation, they need to restrict their physical activity until the myocardial injury and systolic function and arrhythmias is present. When they return to normal, then this activity can be resumed. Now, the second type of the coronavirus vaccines worldwide are viral vector vaccines. As we understood already from the video, the adenovirus, different serotypes of the adenoviruses are used as carriers of the DNA, and then DNA translation and mRNA transcription into the spike protein. And then this spike protein elicits the immune response against the coronavirus. For example, AD26-CoV-2S is the adenovirus serotype 26, which is present in the Johnson & Johnson vaccine. As I told you, Johnson & Johnson requires just a single injection. And it protected against moderate to severe critical COVID-19 with onset at least 14 days after the dose. The efficacy was about 67%, not very great. And at least 28 days after the administration, it was almost the same. So the first data is after 2 weeks, the second data is after 1 month of receiving this vaccine. But when we are talking about the severe critical COVID-19, the protection was higher, 85% of cases after 28 days of administration. And the protection against hospitalization and death at day 28 was 100%. And this is the most important when we are talking about the vaccines. We care mostly about the reduction in hospitalization and death. So it was absolute. This vaccine showed consistent protection across all variants and regions studied. including South Africa. As we are going to see, South African strain, for example, showed very disappointing data regarding the AstraZeneca vaccine, but the Johnson & Johnson vaccine is effective against South African variant, where nearly all cases, up to 95%, were due to infection with this troublesome variant B1.351 lineage. Excuse me. Okay. Next vaccine, we're talking now about the Johnson & Johnson. The next vaccine is the AZD1 or CHADOX1. This comes from the chimpanzee because the adenovirus is the chimpanzee type of adenovirus, or nCoV-19. It is the AstraZeneca, British Scandinavian manufacturer. So this is another vector adenovirus vaccine. And as I said, it uses chimpanzee adenoviral vector containing the surface glycoprotein antigen gene. So as we said several times, the genetic engineering is used to render this adenovirus not infective for the humans. And at the same time, into the genome of this virus, we insert the gene that encodes for the spike protein, which means after injecting the vaccine, there is a DNA transcription, as I said, then an mRNA translation into the spike protein, then spike protein is expressed on the surfaces of the human cells, and the body recognizes it as a virus and starts developing the antibodies. The AstraZeneca also needs 2 doses, 28 days apart. And this is what I mentioned. It was quite fast, the development of this vaccine. Unfortunately, the data on AstraZeneca vaccine varied from one country to another. For example, one dosing regimen showed efficacy of 90% when given as a half dose, followed by a full dose at least 1 month later. Another dosing regimen, when 2 full doses were given at least 1 month apart, showed very disappointing result, only 62% of efficacy. And concerns about the clinical trial implementation and data analysis emerged. These data were not very much reliable because the half-dose regimen was not approved in the study design. So we cannot trust completely this data. Another study showed that after a single standard dose from day 22 to day 90, the protection was 76%. And in the group that received 2 doses of the standard dose, vaccine was higher, efficacy was higher with a longer prime boost interval. So they tried to give the second dose after 3 months compared to the dosage regimen when they gave the second dose after 6 weeks. So the 3 months gave 82% protection as compared to 55% protection if 6 weeks were used as the interval between 2 doses. As you probably heard, the problems with the adenoviral vaccines produced by the Western countries are thrombosis. Thrombosis, different types of thrombosis, mainly the cases were of the cerebral sinus venous thrombosis, starting with the severe headache and neurological symptoms. And this thrombosis was associated with thrombocytopenia. And this was described for both Johnson, which is Johnson & Johnson, and the AstraZeneca vaccine. So interestingly, the Sputnik vaccine, which I will discuss separately, but which belongs only, Sputnik V vaccine also belongs to the adenoviral vaccine or vector vaccines. There is no description of such cases of thrombosis, probably because of the differences in the manufacturing process. But AstraZeneca and Johnson & Johnson were shown to produce these thrombotic complications, and the emergency use authorization of these vaccines now includes a warning that rare clotting events may occur after vaccination, particularly in relatively young women. Okay, who most commonly develops thrombosis after adenovirus vaccine? Women at the age between 18 and 49 years. However, again, as we were talking about the mRNA vaccine, when we compare the risk of this rare adverse effect, which is not life-threatening in most of the cases, and the risk of death from COVID-19, the benefit of administering the vaccine outweighs clearly the risk for the thrombosis with the thrombocytopenia, possibly associated with highly efficacious vaccine. Talking about the thrombosis with thrombocytopenia syndrome, or TTS, which we described for the adenovirus vaccine, it is very rare syndrome, I repeat, and it involves acute venous or arterial thrombosis. It could be either venous or arterial, or it could be a new onset thrombocytopenia, decreased platelet count in patients with no known recent exposure to heparin. Why I'm mentioning heparin here? Because heparin, you know, the direct anticoagulant, it is also associated with thrombosis with thrombocytopenia in rare cases. But here I'm mentioning that even with no previous administration of heparin, such syndrome may develop after the vaccination. The mechanism of TTS is not fully understood, but it appears to be similar to the heparin-induced thrombocytopenia, which is immune-mediated reaction. And this is very rare reaction. In the United States, 2 of 15 persons with TTS that occurred after Johnson COVID-19 vaccination had cerebral venous sinus thrombosis. Cerebral venous sinus thrombosis, CVST, with thrombocytopenia. So majority of thrombosis are in the cerebral venous sinus. So the American Society of Hematology and the American Heart Association, American Stroke Society, have published documents for clinicians to be aware of symptoms, diagnosis, and urgent treatment if TTS is suspected. Diagnosis includes the following 4 criteria. COVID vaccine, Johnson & Johnson, AstraZeneca only to date, 4 to 3 days previously administered. Venous or arterial thrombosis develops, often cerebral, as I said, or it could be abdominal. Thrombocytopenia and positive PF4 HIT score. This is actually a test, ELISA test, which means we can do the laboratory test to confirm the diagnosis. So these are the criteria that should be met to say that this is thrombosis with thrombocytopenia syndrome after the adenovirus vaccine. What are the symptoms that we need to be aware of? Symptoms of TTS that may emerge 4 to 30 days after vaccination with adenovirus vaccines. Severe headache, as I said, unusual acute onset severe headache. There might be visual changes, abdominal pain if there is thrombosis in mesenterial vessels, nausea and vomiting, back pain, shortness of breath, leg pain or swelling, petechia, easy bruising or bleeding. The third type of vaccines is the protein subunit vaccines, or sometimes they are called peptide vaccines. As I told you, we have the Russian vaccine called Epivac Corona, which will be covered later. But among the Western vaccines, we have the NVX-CoV2373 Novavax. It is engineered using recombinant nanoparticle technology from SARS-CoV-2 genetic sequence to generate full-length prefusion spike protein. So the S protein itself is used. It needs the combination with an adjuvant, matrix M, to enhance the immune response to the protein. This vaccine is still under investigation. So far, we are talking about the vaccines that are already in clinical use, the 4 vaccines that we have mentioned so far, but Novavax is not yet in clinical use. It undergoes the clinical trials, and the results of preclinical studies, which means animal studies, show that it binds efficiently with human receptors targeted by the virus. It needs 2 doses 21 days apart. Overall efficacy in the phase 3 clinical trial called PREVENT-19 was quite impressive, 90% or more. And there were no cases of moderate to severe disease in the investigational vaccine group. This is what we care always and always, I repeat, protection against severe disease. In the UK phase 3 trial, when we started having the new variant of concern in the UK, the calculated efficacy against the UK strain was a little bit lower than against the original strain, 86% versus 96%. And regarding the South Africa phase 2B results, the triple mutant variant containing 3 critical mutations in the receptor binding domain and multiple mutations outside the receptor binding domain was widely circulating in South Africa. And even with these new mutations, 60% of efficacy for the prevention of mild, moderate, and severe COVID-19 disease was observed in the study population that was baseline seronegative and HIV negative. Why I'm emphasizing on this? Because when the individual is the HIV positive, it means that he is immunocompromised, and usually they have a poorer outcome in case of any infection. But when we isolate the HIV negative and baseline seronegative, which means that they did not have COVID-19 previously, the protection was quite not bad, 60%. But when we are talking about the protection against severe disease, I'm talking about the South African variant now, there was 100% protection, including all hospitalization and death, which means that Novavax was also quite good in South Africa study. Now, I would like to talk about the variants of concern again. Variants of concern are the new mutants or new variants of the coronavirus. They may reduce vaccine effectiveness, which may be evident by the data that I have presented. And in the real life, we have a number of vaccine breakthrough cases, which means that despite of receiving the vaccine, the person developed the COVID-19, or a very low vaccine-induced protection against severe disease, which is of more concern. That's why the CDC in the US tracks the variant proportions, and they estimated that from January to March, the B117 variant, which was initially detected in the United Kingdom, accounted for a quarter of cases. But in April, it became dominant strain in the United States. But in the mid-June, it remains the dominant strain, now accounting for probably more than 70% of new cases. While the Delta variant on June 15th, that originated in India, and on June 15th, also became the variant of concern in the United States. As of mid-June, the Delta variant was accounting for 10% of new cases in the US. I'm talking about the US data just to show you how the countries like US and Britain, they conduct the surveillance. And also in Russia, the Delta variant currently is the dominant variant or dominant mutant, particularly in the big cities such as St. Petersburg and Moscow. Other variants of concern, as I mentioned, include the B1351 or 501Y.V2, which is the South Africa, and B1128, renamed B1, which is Brazilian, and it was first discovered during routine screening at the Tokyo airport for travels from Brazil. And the change of the B117 variant that includes a particular mutation, E484K. And now this variant is called B117 plus E484K, also is of future concern. Also, there is Californian strain in the United States, another variant of concern, and it is called B1427 and B1429. They both emerged in California. So let me make a break of 5 minutes, and now it's 11:45. We'll resume our lecture at 11:50. I will mute myself and stop. All right, guys, we go on. We discussed the variants of concern and variants that have emerged in the UK and South Africa in late 2020 have multiple mutations, as we could see in the table previously. These mutations occurred in their S-glycoproteins, which is spike protein. And this is the key target of currently available vaccines. That's why there is a concern that vaccines will lose their efficacy. And this is what's happening in some of the cases. Now, regarding Moderna, I have already mentioned that they are trying to produce the booster dose because they have shown 2/3 reduced neutralization of vaccines against the B.1.351 variant in vitro, and other in vitro studies also compared the sera of neutralizing antibody titers from participants in vaccine studies. They described that the sera from the Moderna vaccine showed no reduction in neutralization of pseudoviruses bearing the UK variant and the South African variant. However, the other studies contradict these results. They show that there was good protection against the new variants and most other variants. So this is not very well defined point. And particularly because now we have the Delta variant B.1.617.2 that rapidly spread in India and becomes now the predominant variant in many countries. This variant is shown to be almost 7-fold more resistant to neutralization by sera from COVID-19 convalescent and mRNA vaccinated individuals. And here I would like to clarify this point better. Being a 7-fold less effective for the vaccine is not a problem, but for the convalescent individuals who recover after COVID-19, this may be a trouble. And this explains why the people who sustained the COVID-19, they become reinfected with the new mutants because the titer of antibody in their blood is not sufficient to protect against the new mutants. For the vaccine, this is less troublesome because the even 7-fold decrease in the titer of the antibody still offers sufficient titer of antibodies to be protective against the new variants. I hope you got my point. So this is the information that I have told, that Moderna is now working on the booster, and actually this is the repetition of what I said. I'm sorry about this. And about the booster, I've already talked in the beginning of this lecture. Regarding the Novavax, I have already also discussed that against South African strain, the efficacy was much less than against the original strain. And regarding the Johnson & Johnson variant, also it is losing its efficacy against the Latin American South African strain. These are protection or efficacy numbers much less than against the original strain, but still it is 85% effective in preventing severe disease and complete protection against hospitalization and death in all geographic regions. That's why I know a couple of my friends, there are a couple of physicians, they traveled from South Africa to the United States to get the Johnson & Johnson vaccine to protect themselves against South African strain, which is the B.1.351 lineage. Exactly. Regarding the AstraZeneca vaccine, as you probably heard in the social media, when this vaccine was tried in South Africa, the very low efficacy of the AstraZeneca vaccine, only 22%, was detected. And these results prompted South Africa to halt enrolling the vaccine participants in early February. It is not useful against their strain. And in England, 2-dose effectiveness was reduced from 66% to 59.8% against Delta variant. But I do not see that this is a very dramatic decrease, 66 to almost 60%. But still, we can observe the decline in the effectiveness of the vaccine. And after this brief overview of the Western vaccines, now I will talk about the Russian vaccines. And I apologize that the text is in Russian. Hopefully, most of you already are at the senior years and you can read Russian, but I will comment on the vaccines that are being developed and used in Russia. Starting with the first COVID-19 vaccine, Sputnik V. As you know, this vaccine was the first one to be registered in the world. The trade name is Sputnik V, but the scientific name is GAM-COVID-Vac. It is developed by the National Research Center of Epidemiology and Microbiology, named after Gamaleya, belonging to the Ministry of Health of Russia. And it is produced in a number of companies, including the Gamaleya institution itself in St. Petersburg and some other cities in Russia, including Ufa. You know, the Ufa, the Biopharm Standard Factory, is also producing the Sputnik V vaccine. This vaccine has passed all 4 stages of the investigations. One was preclinical, which means in animals, as I told you, and 3 clinical stages, which means it was on humans. And it was registered in August 2020, as I told you, the first registered anti-COVID vaccine worldwide. And it is similar to other adenovirus vector vaccines. In what respect? That it consists of 2 components. It is very similar to AstraZeneca vaccine, where we have 2 recombinant adenoviral vectors, but these two doses are different. The first vector is different from the other vector. As I mentioned, for the other adenovirus vaccines, we have the genetic engineering, producing the genetic material with the insertion of the gene encoding for the S protein. And this fragment of genetic material is safe for the human, but is able to produce the stable antibody and cellular immune response against the virus. You may ask, what are the possible side effects? During the first or second day after injection, there might be short-term general and local reactions like chills, elevation of body temperature, but not exceeding 38.5, myalgia and arthralgia, which means the pain in the muscles and joints, fatigue, headache, pain at the site of injection, and redness. Usually this subsides during the 2 to 3 days, and the safety has been completely investigated. The technology of using of adenoviral vectors as vector vaccines is safe and effective. And the data of phase 3 of the clinical trial of Sputnik V was published in one of the most reputable journals, which is the Lancet, in February 2021, showing safety of this vaccine. Regarding the effectiveness, it is very impressive. Currently, it is 97.6%, and it is recommended for all adults above the age of 18 years. The second Russian vaccine that is also in use, called Epivac Corona. As I told you, it is very much resembling the Western Novavax vaccine. It is based on the peptide antigens and also used for the COVID-19 prophylaxis. It was produced by the Federal Budget Institution, scientific institution called Vector. And now it is produced by Vector itself and some other platforms. It is registered in October 2020. Currently, the clinical investigation has been completed based on the 150 participants, all the 60 years and older. And this means that this type of vaccine is safe for this age. Elderly people can use, also similar to Sputnik V, the Epivac Corona vaccine. And currently there is an ongoing clinical trial based on 3,000 participants, volunteers at the age of 18 and above, based on 8 clinical centers in several cities, including Moscow. The purpose of the ongoing trial is the acquiring of additional data regarding the safety and effectiveness of the vaccine and about the characteristics of the unwanted reactions. In June, very recently, at the end of June this year, the registration certificate has been changed, and now the new form and the new dosage form was approved, which is glass syringe. Now there is around 3 million doses in the market, or in the use in Russia, which allows us to vaccinate more than 1.5 million people. What about the composition of this vaccine? As I told you, the composition is the protein, which is the carrier, and artificially synthesized peptides. And these peptides are small fragments of the S protein of the new coronavirus. So there is no actually virus or its parts or genetic material. That's why basically we do not have the side effects from the vaccine. It is very areactogenic, which means very small or almost no allergic reactions, and only short-term pain and some increase in the body temperature may be observed. The vaccine has showed its safety on volunteers, safety and also effectiveness, as I said, and currently it is recommended to any adults after the age of 18. As I told you, this is a very areactogenic vaccine, meaning that immune load is minimal, and that's why it is particularly recommended for the people who have chronic diseases such as coronary artery disease, diabetes mellitus, hypertension, of course, outside the exacerbation, there should be remission of the disease to receive the vaccine, as for any other vaccine. The vaccine very well boosts the immunity, and particularly after the coronavirus infection, it can be safely used. The contraindications are only hypersensitivity to hydroxide aluminum, which is one of the adjuvants, or severe allergies in the past, reactions to the previous doses of vaccine, primary immunodeficiency, cancer of the blood, and other tumors. The Epivac Corona also needs 2 doses, 3 weeks apart, and the immunity will be on the 42nd day after the first dose. The third Russian vaccine is Sputnik Light. It is also developed by the same institution as the Sputnik V, National Research Center of Epidemiology and Microbiology, named after Gamaleya. And now it is produced by a number of manufacturers, including the UFA Farm Standard vitamin factory. And of course, the center of Gamaleya itself. On the 6th of May this year, the Ministry of Health of Russia has registered this vaccine. It has already completed 3 out of 4 clinical trials, including the preclinical, which is animal, and clinical. And currently, as you can guess, it is in the 3rd phase of the clinical trials. And there is a global investigation launched by the Gamaleya Institution, and it started on 21st of February 2021. And this 3rd phase has 7,000 participants, including Russia, United Arab Emirates, Ghana, and some other countries. So we'll have hopefully very soon the international results of using the Sputnik V vaccine. Yes. By the way, I forgot to mention that Sputnik V itself is used already, is approved in about 50 countries worldwide. And this, by the way, includes Jordan, and in Jordan, it was successfully used in several tens of thousands of people. Going back to the Sputnik Light, the Sputnik resembles a Sputnik V. And actually, it is exactly the copy of the first component of the Sputnik V. It is produced by the biotechnologic way, not using the virus itself of the SARS-CoV-2, but as you already know, using the adenovirus as the vector. So like Sputnik V, the Sputnik Light produces the humoral and cellular immunity regarding the coronavirus infection caused by virus SARS-CoV-2, similar to Sputnik V, but instead of 2 components in Sputnik V, in Sputnik Light, we have just the first component. This is the difference, and that's why the name Light. What adverse effects are possible with Sputnik Light? Similar to the other vaccines, even from the childhood, you remember that any vaccine can produce the general and local reactions, such as we know, chills, fever not exceeding 38.5, pain in muscles and joints, fatigue, headache, pain at the site of injection, and redness. But usually these are totally disappearing, these adverse effects, within the 2 to 3 days. The Sputnik Light was shown to be safe, including those people who had preceding immunity to SARS-CoV-2. I mean, those who have recently sustained the infection. The effectiveness of the Sputnik Light is 79.4% after 28 days of infection. To whom it is recommended? Priority is for the repeated vaccination. You remember we were talking about the booster recommended by the Moderna against new variants. But now for the Sputnik, those who received the 2 doses of Sputnik, they can receive the 3rd dose of Sputnik Light. And also it can be recommended for the individuals who sustained COVID-19 for the purpose of facilitation of immunity in the pandemic circumstances. And it is currently approved at the age between 18 and 60 years. And finally, the 4th Russian vaccine is Covivac. Covivac is produced by the Federal Scientific Research Center for the development of immunobiological preparations named after Chumakov of the Russian Academy of Sciences. It is officially registered in February 2021, and in June there is a permission or approval of the 3rd phase of the clinical trial, and currently it is ongoing, and it is based on 30,000 individuals. Other vaccines that are in the future will be St. Petersburg vaccine of the FGUP, SPB, NII, BC, FMBA, Russia. There is no name yet because it is investigational. So it is still under the development, as I told you. And the platform of this new vaccine is actually aiming the cellular immunity. If we are talking about the previous vaccines, they were mainly aiming the antibody response. Now, here with this vaccine, they will be targeting the cellular immunity. And they will be targeting those conservative proteins of the virus that are less subject to mutations. Very recently, last month, the preclinical trials were completed, and they have shown safety, immunogenicity, and protective ability or potential of the vaccine. Documents are now prepared and submitted for the clinical trials. At the end of June, these trials have been approved, and they're planned to be in the nearest future. So the details about the composition of this vaccine is not revealed, probably because of patency or intellectual property things. Now, also the adenovirus-based vaccine, which is developed by the CanSino Biologics, the Chinese company, is probably to be produced by Russian companies. The company from China, CanSino, received the permission of the Ministry of Health for the participation in the international investigation of the third phase to detect the efficacy and immunogenicity of the vaccine. And the part from the Russia that participates in the third phase of the clinical trial is the Russian company Petrovax. So this was the brief overview of the Russian vaccines in use. 4 vaccines we have mentioned: the Sputnik V, the Sputnik Light, the Epivac Corona, as well as the Chickenpox vaccine, Covivac, and the other vaccines which are under investigation. Now, I would like also to briefly mention at the end of today's lecture the other investigational vaccines worldwide. We have a number of DNA-based vaccines by Inovia Pharmaceuticals and mRNA-2-dose also vaccine by the CureVac, the recombinant protein adjuvant vaccine by Sanofi and GSK. They have completed phase 2 and they are starting now the phase 3. The S-trimer vaccine by Clover and Dynavax, phases 2 and 3 trial are ongoing. It also requires 2 doses, 21 days apart. The VLA vaccine, which is an activated whole virus vaccine in the UK, is investigated by Valneva and Dynavax. And UB612 multi-top peptide-based vaccine by the COVAX United Biomedicals, also undergoing phase 2 in Taiwan, phases 2 and 3 in Brazil. And they are, you see that some companies are merging to form a new company. Halovax vaccine, which is produced by the Massachusetts Center, which is a self-assembling vaccine platform. Nanoparticle SARS-CoV-2 vaccine, Urovax, self-assembling protein nanoparticle platform technology. PDA0203 utilizes Versa immune T-cell activating platform. Probably this is similar to the St. Petersburg vaccine. Maybe it is actually our vaccine, but we do not have the details here. COVID, LP, Medicago, and GlaxoSmithKline, which is combining the recombinant coronavirus virus-like particles with GSK adjuvant system. Phase 3 trial initiated and ongoing. Covaxin by the Bharat Biotech and Ocugen. Polvarion, an activated candidate. This is Indian platform, but also they collaborate with the US market. And an activated COVID-19 vaccine, Sinopharm. Actually, this is not investigational vaccine. As I told you, I received this vaccine and more than 1 billion doses were received now by people worldwide. And it is already approved in China and Saudi Arabia and Jordan, for example. And the data are not bad, 86% efficacy. And WHO approved this vaccine for the emergency use in May 2021. Sinovac vaccine, which is coronavac vector, adenovirus vector vaccine, has limited use in China. The trial in Brazil was not impressive, efficacy just 50 to 90%. Turkish, however, trial gave brilliant results, 91%. So probably depends on the strain. And Sputnik V, we have discussed in all details already, and it is not investigational. It is very well known in Russia. As you can see, we have a number of other vaccines. The new approaches are vaccines targeting nucleocapsid and outer spike protein, which can activate T cells and antibodies against SARS-CoV-2. mRNA-based vaccines, further by Sanofi and Translate Bio. Adjuvanted DNA vaccines. Basically, the same 4 types of vaccines we're talking about. So as you can see, big international companies are all competing, and they are conducting different phases of trials. What I would like to state at almost the end of the lecture, that very attractive approach is to give the non-injectable vaccines. In the nearest future, because now they are investigational, in the nearest future, we will be able to give the drugs intranasally or probably orally. One of these vaccines is HAD5 T cell, targeting also nucleocapsid, because nucleocapsid protein undergoes less mutations. Also outer spike S protein to activate T cells and antibodies. And now there are phases 1 and 2 trials. Intranasal COVID-19 vaccine by Altimmune, 1 or 2 intranasal doses. That phase 1 trial was started. The VXA-CoV-2, 1 oral vaccine, which is based on the adenovirus platform. So basically the same platforms, but they are trying to change the route of administration. Why is that? Because probably I have to mention here a very essential point, that when we give the vaccine by injection, the immune response, it develops only in the systemic circulation. And okay, we protect our bloodstream, we protect our lungs, maybe our inner environment against the virus by the inner immune response. But what is the gate for the viral entry? It is the nasal and oral mucosa and upper respiratory tract mucosa. When we give the vaccines, traditional vaccines, I mean by subcutaneous route, we do not eliminate the virus that may already be present in these external areas of our body. So that's why the vaccinated people should continue wearing masks, because they may still infect the others. They may still spread the infection. And that's why probably the pandemic is still ongoing. So very attractive is to give the vaccine topically on the nasal mucosa, as we said, intranasally, or to the oral mucosa, enhancing the local immunity against the virus and eliminating virus from these surfaces, preventing its penetration to the lower parts of the respiratory tract and into the systemic circulation. Next is transdermal microneedle. I remember when I was a school child, this transdermal microneedle or analog was used during cholera, and also for a short time it was used against influenza. This transdermal microneedle COVID-19 vaccine is produced in the States, and a number of universities and companies are working. They started the testing in preclinical settings in mice only. Attractive about this vaccine that it is easy to mass produce and store without refrigeration. We have major problem in transportation of these vaccines, right? Another vaccine, intranasal live and attenuated. This is what I was talking about. We are concerned about the mucosal immunity, surface immunity, production of immunoglobulins A in nasal cavity, and systemically circulating antibodies are also produced. Triple antigen oral vaccine, Oravax, is planned for the mid 2021. I mean, they will begin the clinical trials, and another oral vaccine, Esperovax, has just gained funding for the research. So these are the future steps. We hope that we will have more and more vaccines targeting new variants or polyvalent vaccines, like we have with pneumococcal vaccines. We can target many mutants at the same time. By now, concluding this lecture, I would encourage you to go and vaccinate yourselves as soon as you can, because as you could see from this talk, the vaccines are effective and safe. Thank you. These are the references that I have used overviewing the vaccines. Thank you very much. If you have any questions, still, you can maybe communicate through WhatsApp or other messages. If you have any questions, if you have any comments, I would like to, address them. Thank you very much for your listening.
Thank you for your lecture. Thank you.
You are welcome.
Dear students, you're welcome with your questions.
I think they are muted. Maybe you can allow them chance by
just a second.
unmuting them. If you'd like, we have remaining 10 minutes. If you have any questions, any concerns, any unclear issues, you are welcome. I think they are still unmuted. Muted, sorry.
Just a second. So there's some problems with. Сейчас, секундочку, хорошо?
Ага, я себя уже озвучила.
Так, а вы можете тоже, да, включить, потому что что-то у меня тут.
Их сейчас я попробую. Participants, maybe participants. Нет. Так,
Я думаю, нам кто-то там. Так, сейчас я сделаю разрешение. Можно, я думаю, попробовать ребятам задать вопрос.
Можно я спрошу, когда я видео демонстрировала, их звук было слышно или только мои комментарии?
Только вы.
Жаль, я тут пробовала как-то озвучить.
Мгм.
Hello, good afternoon.
Ну,
Yes, yes, hello.
Yes, ma'am, can you hear?
Yes, please. Do you want to ask me a question?
Yes, ma'am. I wanted to ask if we took the COVID vaccine, how long does the immunity works?
You
I
know,
mean,
because, yes, this is a question that disturbs many, many minds. And because the vaccines started being used worldwide only at the end of 2020, let us say December 2020, January 2021, the investigations are still ongoing. And if we are talking about the clinical trials that preceded this wide use of the vaccines, they are guaranteeing now at least 6 months of protection. Further data are monitored and being collected, and with time will accumulate more data. Now they are saying minimal protection, but hoping that memory B cells and cellular immunity will offer the long-term protection as well.
okay, ma'am. Thank you.
You're welcome. Other questions, please?
Hello, ma'am.
Good afternoon, ma'am.
Hello, good afternoon.
Ma'am, if patient is recover from COVID positive and he become negative, after how much month he can take vaccine, ma'am?
Recommendations vary. For example, in Russia, it was recently 6 months after COVID-19 and after the antibodies against coronavirus become negative. But I believe this has been changed recently, and also in Jordan, it was initially 3 months after you sustained the infection. Now they are saying you can go and vaccinate. And as you could see, I was displaying the CDC, which is the American recommendation. As I mentioned at the beginning of this lecture, they are saying that immediately after finishing your isolation period, you can go and vaccinate itself, unless you received the reconvalescent plasma, or monoclonal antibodies, after which you have to wait for 90 days.
Ma'am, if he has antibody also, then also he can take vaccine.
If you received the monoclonal antibodies, in the my next lecture, I will be covering the treatment of COVID-19, and you will hear about the specific anti-S protein, anti-spike protein monoclonal antibodies. This is what they mean, monoclonal antibodies. If they were used for your treatment, which is most probably in the United States, and also reconvalescent plasma, which is used in many countries, I believe, including Russia. If the reconvalescent plasma was used for your treatment, which means that you were severely ill and in the hospital, you received the plasma from the convalescents. What is the meaning of the convalescents? Those who have recovered from the infection, and they gave their plasma as donors, and then it was injected to you because you were severely ill. Only in this case, you have to wait for 90 days.
Okay, ma'am. Thank you, ma'am.
This is international recommendation. Because I told you, different countries, they use different vaccines. And regarding they are mostly using mRNA and adenovirus vector vaccines. This spreads for these types of vaccines.
Ma'am,
Yes, please.
ma'am, here in Ufa, it's not required to go for an antibody test if you are positive before for taking vaccine.
Before receiving the vaccine?
Yes, ma'am. Like if someone get positive and he want to take vaccine, it is not required to go for antibody test.
No, it is not required.
Okay, ma'am. Okay.
Yes, other questions?
Excuse me, ma'am. Good
Yes.
afternoon. As we know, the Delta variant of the coronavirus, is this a mutated version of the coronavirus?
Is this the?
Mutated version.
Yes, it is
Does the-
mutated, and there is more than one mutation, which resulted in the more rapid spread. More people are infected. If with the original Wuhan variant, there was a ratio like 1 to 1 or 1 to 2. One infected person could infect 1 or 2 more other people. Now with this variant, the ratio is almost 1 to 8. So more widespread, and also, as I told you, more resistance to the monoclonal antibodies that I mentioned that are used in the treatment in some countries, and possibly there is a poorer response to vaccine. That's why I repeat, some companies are thinking about the booster dose, which is actually the third dose of the vaccine.
So, ma'am, does a single dose, single vaccine can capable to cure it? Because
Yes.
it shows the mutation against the vaccines which are created by doctors.
Yes, this is a very important question. You know, since the Delta variant is quite new, we do not have exact information about protection by the vaccines. But as I told you, in vitro data, when you give the vaccine and then you mix the plasma of the vaccine recipient with the virus and see neutralization of the virus in vitro. And you compare that with the neutralization produced by the plasma of the reconvalescent. Still, the protection is offered by the vaccine 10 times higher than the protection after the infection. What I'm trying to say that, okay, even if you recently had the COVID-19, you are less protected compared to when you recently had the vaccine. Okay, the vaccine also has less response, but still, because of the titers of antibodies are much higher than after the disease, you are protected to more extent. And what is of our concern, I again repeat, is that you are protected mostly against the severe disease, hospitalization, and death. This is what we care about. It's not a problem if someone gets a mild to moderate disease. The problem is when somebody dies or is, handicapped after the infection, because his lungs are not working, because he has neurological consequences, and so on. So go ahead for the vaccine. It is much safer than having the COVID-19, briefly saying.
Okay, ma'am.
Hello, ma'am. Can I ask a question?
Ma'am,
Yes, please, Asia.
Ma'am, I heard like if after vaccination, some people got COVID-19. So my question is that possible even, or it's not possible?
Yes, as I said in my lecture, there is no absolute protection. Even the best vaccine, let us say Russian Sputnik, is 97% protection, which means that still there is remaining 3% chance of getting infection. But what I'm saying, this infection will be mild to moderate, because the protection against severe infection, hospitalization, and death is 100%. So if you get the disease, you will survive. Okay.
Thank you, ma'am. I get it. Yeah.
Do we still
excuse me, ma'am.
or do we have
Ma'am, I have
limitation
a
of time
question.
or not? I'm asking, excuse me, I'm asking the Dean's office. Do we have time to take additional questions, or we are limited?
Of course we have.
Okay, thank you. Go ahead, please. Thank you very much.
Excuse me, ma'am. I have a question, ma'am.
Yes, sure.
Ma'am, what is
Please.
the major cause of mutation in the new variant, ma'am?
A very, very good question. Here we come again to the importance of the vaccination. The more the virus persists in the population, the longer we are staying unvaccinated, the higher is the chance of mutation of the virus. The more quickly we cover the whole population, or at least majority of the population. Now the target is not like previously. Previously, they were saying 60% of herd immunity or population immunity is enough to protect against the pandemic. Now they are targeting with new mutants. They are saying, the authorities are saying 80% of population should have the herd immunity. Herd immunity means those who have had the disease recently and have the vaccine. So if altogether this herd immunity will reach 80%, then we will be free of mutations. But if the majority of the population is not vaccinated and the virus stays for longer time, the longer the virus stays, the higher chance for mutations. And also this includes people who have severe forms of the disease and they stay a long time in the hospital, they stay a long time in the ICU on mechanical ventilators, you know, on the ECMO, which is extracorporeal membrane oxygenation. These severely ill people, they have their virus, they did not eliminate their virus, right? So the virus persists in their bodies for a long time. And these are the sources of mutant. And the same we may say about the immunocompromised people, because their immunity cannot struggle with the virus. They also, you know, produce mutants like cancer patients, like other immunocompromised patients. And also, unfortunately, the use of monoclonal antibodies against the spike protein, which I will cover in my next lecture, their wide use in the United States and in the other countries, and also use of convalescent plasma, it also resulted in the development of the mutants. So the solution is to get the maximum vaccination of the population.
Okay, ma'am. Thank you.
All right. Okay. Other questions, please? Do you have other questions, guys?
No, thanks.
It seems that Salama wants to ask. No?
I said no, thanks.
All right, then we will conclude today's lecture. Thank you very much, and see you after tomorrow. Bye-bye.
Yes, ma'am.
Thank
До
you.
свидания.
Thank you, ma'am. До свидания, ma'am.
До свидания, ma'am.
До свидания.
До свидания, ma'am.
Okay.